This is the lecture that covers Chapter 10 from your textbook on complex research designs. I've made a few small modifications to the slides as they exist on the Canvas page, but there's no new information, just a couple of additional images that I wanted to share and a little bit of reordering. So let's go ahead and get started. I want to begin, as you know, as typical with these chapters, I want to talk about some actual research and how it relates to the constructs that we're discussing in this chapter on more complex experimental designs. So if you remember previous lectures, we've talked a lot about how there's this really important task in science of taking concepts, constructs, hypothetical things that you can't touch or examine in the real world that exist sort of in an abstract way, and operationalizing them and being able to measure them and look at them in a scientific way and to develop data that can then be tested statistically. So converting them into numbers and measurable phenomena. So that's an important process, and it's important that we do that for all key variables that are under study. So let me give you a couple of examples and work up to an example of a more complex research design that we're going to come back to over the course of the chapter. So there is a lot of groundbreaking research on facial expression of emotion in the last few decades, and specifically Ekman and colleagues in some of their research have shown that there's a universality of human facial expression of emotions, that it seems to cross cultures, that there is some cultural variability, but that essentially it seems to be a very human phenomenon. So in this conceptual definition of emotions, as in their theory, emotions are response systems, right? They're things we've evolved and developed to respond, to read and to understand and to how to respond to stimuli in our environment. And so they say that facial expressions are a critical medium for communication of emotion in human beings. And what Ekman developed starting in the 90s is what's called a facial action coding system. And what he did was he came up with operational definitions for all the various facial expressions of emotions. And he cataloged various things that the human face does, that our eyes do, that our expression does, that communicates emotions that seem to be universal across culture. He's replicated this stuff, and many others have, and found that it consistently holds. And they identify 7 discrete emotional expressions that humans use and have the capacity to recognize across culture. And we'll go through each of those in the next slide. So these are the operational definitions that he developed, and he argues these are essentially, exclusive from one another, and that they capture the majority of what we communicate through our faces as regards emotion. Happiness, sadness, fear, anger, surprise, disgust, and contempt. And as you can see, what he's done is developed a very specific operational definition of what the face does to express these emotions. If you sit with a mirror or with your phone for a moment on, as if you're taking a selfie, and you look at these descriptions, the corners of the mouth lift into a smile, the eyelids tighten, the cheeks rise, the outside corners of the brow pull down. So if you essentially try to mimic these descriptions, what you'll find is that you're presenting a relative, facsimile of that emotional expression. And it was really through careful study and examination of lots and lots of different subjects that they found these consistencies. But anyway, the idea is this is a really good operational definition of something that is relatively abstract, which is how do we convey emotions through our faces. Now, the study that we're going to discuss in more detail in this chapter is the work of Adams and Kleck from 2003. And this is where they wanted to study facial expressions of emotions based upon the previous research from Ekman, but they wanted to add something really important. They had a theory that said that the facial expression of the emotion, that universal communication, is very important, but that the direction of the gaze of the eyes is also very crucial. Now, I've been making these lectures for over a year now, and I've been doing a lot of Zoom meetings, as you can imagine, and doing therapy, and I have to force myself to remember to look at the camera lens, okay? I have a tendency to want to read what's on the screen or to look at my notes, and even when I'm with patients, I'm always taking notes during sessions, and I have to be really careful to make sure that I'm not looking at their image on my screen, but as I am now, that I'm making eye contact with the camera itself. So there is an important distinction about eye contact and what it conveys in relationship to those emotions. So they clearly feel and have good evidence to support the idea that these are distinct from one another, right? We can all generally agree that from an operational definition, the look of an emotion on your face and where you're looking are what they call orthogonal or mutually exclusive things. And they argue that it can vary across two basic levels, and that each of them can then be represented as an independent variable. So there's now we now have two independent variables in hand that we want to talk about and examine in the form of an experiment. We want to see how both of these things potentially have a causal impact on the dependent variable. So what you have is you have now two independent variables, which is which emotion is being expressed on the face, and which direction is the participant's eye gaze looking at. Is it looking at the person or averted? Is it looking directly into the eyes of the person viewing the image, presumably, or is it looking askance, averted from the viewer? So that's one independent variable with two levels, averted or direct eye gaze. And the other is which expression is being conveyed on the face, and they really want to focus on anger versus fear as these important evolutionary emotional constructs. So they had to design a study that was able to capture both of those independent variables within the design, and looking how it then has an impact on people's ability to interpret it. And that's their dependent variable. So this is just an example from their article of certain emotions that are based upon the Ekman 7 established universal emotional expressions. And what you're seeing is that this is the same person mimicking the same facial expressions, but that they've gone in digitally and moved the eye gaze. So this is not two attempts. This is actually one attempt, and they just changed the direction of the eyes digitally in Photoshop. So what these researchers are utilizing, and what this chapter is really about, is what we're going to call multifactorial designs. If you remember back 2 chapters ago in the chapter on experimental design, we talked about single factor experiments, and we really were only talking in that context about single factor experiments, one independent variable as it influences one dependent variable. In this case, we're now talking about introducing multiple factors, multifactorial. So the factor is essentially the independent variable. So a single factor study is a single independent variable study. But now in more complex designs, we introduce multiple independent variables, and we call those factorial or multifactorial designs. Anytime you're varying 2 or more independent variables, then you meet this definition. And so you can look at the presumed or the hypothesized effect of each independent variable on the dependent variable. You can isolate it and test them as standalone. But then you can also isolate something very interesting, which is called an interaction effect, which is how do these 2 independent variables in tandem selectively with one another affect the dependent variable? Is there some important relationship there that we're missing that's really not due to any of them in a vacuum, but only as they interact with one another? So this can be a really efficient design because it allows the researcher to study more variables in a single study, obviously. It's also very good for understanding complex phenomena because most things that we study in psychology, most things about human psychology or human emotion is, we would argue, complex, right? It's not simple. So we know that most psychological phenomena are not the product of only one influence or one causal factor. A factorial study really allows the researcher to look at multiple factors and how they relate and interact with one another on a phenomenon. So as I mentioned, the Adams and Kleck study, that's an important example of a 2-factor multifactorial design. The 2 independent variables being the emotional expression, anger versus fear, so 2 levels to that independent variable, and the second independent variable, which is direction of eye gaze, direct or averted. And that's a 2-level second independent variable. So we utilize a certain notation to talk about multifactorial designs, and it has its own sort of like a nomenclature, okay, that allows us to understand very quickly and communicate very quickly what type of multifactorial design it is. So this numbering rotation is interpreted on 2 levels. And so I'm going to be very careful about my language here. The number of numbers in the designation tells you how many independent variables are included in the study. The value of those numbers indicates the number of levels for each independent variable. So you can think about it this way. In the Adams and Kleck study, we have 2 independent variables, okay, and they each have 2 levels. In the nomenclature of a factorial design, we would call this a 2 by 2 factorial design. There are 2 numbers and each of them has two independent levels of the independent variable. So there's two independent variables, and each of them has two levels. Now, to maybe clear this up a little bit and hopefully make the connection here, imagine for a moment if they decided to use three different emotions, right? Not just anger and fear, but let's say disgust. Then we would probably designate that as a 2x3 multifactorial design. We still have two independent variables. There's a 2 and there's a 3. But those numbers are telling us how many levels there are to that independent variable. So the classic Adam-Kleck study is a 2x2, and you can have anything. It gets more and more complex. If you have lots and lots of independent variables, which occasionally will happen, you might describe it as a 2x3x2x2 if you had 4 independent variables, right? And they each had 2 levels except for one, which had 3. So it can be utilized to describe very quickly and very succinctly the basic structure of a multifactorial design. If you want to know how many conditions there are going to be in this study, how many conditions that a person who's participating could be randomly assigned to, for example, you simply play out that mathematical formula. A 2x2 means there are 4 cells, right? And so that should make sense if you think about it for a moment as almost like a matrix where it's crossing over. We have 4 cells that are created. We have anger in the Kleck study, Adams and Kleck study. We have anger with averted eye gaze, anger with direct eye gaze. We have fear with averted eye gaze, and we have fear with direct eye gaze. So you create this overlap that creates 4 possible conditions. So by playing out that nomenclature, that 2x3 or 2x7, whatever, for example, a 2x3 would have 6 conditions. A 2x7 would have 14. Sorry, 2x7, yeah, but that would be a pretty rare design. You wouldn't usually have 7 levels of your independent variable, but you get the idea. You simply just play out the formula and then you have your answer as to how many possible conditions there are. Now, this is where I think these constructs start to overlap and things can become slightly confusing, so I'm going to go through this very, very deliberately. In the previous chapter, we talked about between subjects and within subjects designs, right? The idea that in a between subjects design, remember, each participant experiences only one condition of the independent variable. One level, at that term, we're only talking about single factor designs, one level, but we're now going to use the term condition, right? Some combination of variables. In a between subjects design, the participant only lands in one of those boxes of that matrix, so to speak. They're only assigned to one condition. In a within subjects design, every participant experiences all levels of the independent variable. So for a moment, I want you to think about this, that a multifactorial study, one that involves more than one independent variable, could be mixed in the sense that the person might experience only one level or one level of one independent variable, but all levels of the other. What you then have is a mixed between and within subjects design in a certain way. It's in reference to which independent variable you're referring at the time. So in the Lu study, where they had the Chinese students that were trying to learn English, and they had some who exercised while they learned versus those who remained sedentary, right? That's a between subjects independent variable. Those people were either in one or the other. You were either riding the bike while you learned English words, or you were sitting in a quiet classroom while you did it. So that's a between subjects independent variable. We talked about the Stroop test, right, as a measurement of inhibitory control and cognition. That's where, if you don't remember, that's where you're presented with the spelled out names of colors, but they're printed either in the same color that they are written as, or they're printed in different colors. If you go back and look at previous lectures when we talked about the Stroop test, you can refresh that. But the Stroop test is a within subjects design. It doesn't even make sense with the Stroop test to not present the congruent and the incongruent test. That's where you get your actual score. Your dependent outcome comes from them experiencing both levels. So what you've got now is examples of within and between. You could basically create an example of a mixed factorial design, which could be that you could give the Stroop test to the subjects in the Lu study, right? But that would be a within subjects independent variable, right? Because everyone would experience the incongruent and the congruent. But you could also have them still separated into people that are riding the bikes and people that are sedentary, which would be in between subjects. So that's just an example of how you can have a mixed factorial design. Things can get very expansive very quickly. And so there's always this pushback between, I want to study everything. I want to make sure that I'm, examining all aspects of my theory. and running into trouble because the study starts becoming unwieldy. You start having too many different cells to fill with participants. It becomes more difficult to get enough subjects to have enough statistical power to detect those hypothesized relationships. This goes back to our last discussion about type 1 and type 2 errors. So you're in a situation where you have to think all of the pros and cons through in terms of how many factors to have in your multifactorial design, and then how many levels to have within each of those variables. But this mixed factorial design might be a good way to decrease the number of participants that you do. If you can include at least one within subjects variable in your design, well, then you're getting an additional point of data at every level of that independent variable from each individual. So mixed factorial designs allow you another technique, another tool for creating an elegant design for your research study, to look at things in a way that makes sense based upon your theory and based upon your resources, what you can do. So in a sense, you're adding an independent variable to a single factor experiment. So when we talk about single factor experiments, as I said, that's one that has one and only one independent variable that has two or more levels. You have to have at least two levels to compare something, right? In a multifactorial experiment, we're adding at least one additional independent variable that's incorporated into the research design. There is some examples from your book that I won't get into too much detail, but they talked about Rowe, Hirsch, and Anderson. They did a study in 2007 that looked at mood induction, being able to induce a mood in somebody, versus that happening through two different types of cognitive exercises, verbal or visual. What they found from this interesting design was that a happy mood was improved performance for generating remote verbal associations. If you were in a happy mood, you did better at verbal tasks. But if you tried to perform a visual task and you were happy, you actually did worse. It was really interesting. And they saw that there was actually this interaction between the mood and what the type of thinking you were doing, what type of cognitive task you were engaging with. So it's just a way to sort of look at the combination of two. As I mentioned earlier, what's really interesting about utilizing multiple factors is not just seeing the potential for a cause and effect relationship between each isolated independent variable and the outcome, because if you think for a moment, you can always just collapse across that second independent variable and just look at whether or not the first one has an effect. You can also do the same thing, collapse across the first one, all the subjects, and just see if the second one has an effect on the dependent variable in both cases, right? But you can look at how they interact. Without this type of design by Rowe and colleagues from 2007, you would not understand that yes, your happy mood does affect your performance, but it affects your performance depending on the type of task that you're engaging with. And you only learn that type of knowledge about your theory by designing a study that allows you to examine the interaction of the two independent variables. So this is what they found in the in this study. That basically these are their two independent variables: mood induction and cognitive task, right? So mood induction is basically you utilize a technique to sort of try and bring about some semblance of this emotion within the subject before they then go on with the task. So if you are induced to be happy, for example, you did better on verbal word associations. Probably we think of this as sort of left hemisphere of the brain type language-based stuff. When you're engaging the right hemisphere of the brain, you're doing a visual task, you actually had diminished attention, diminished performance when you were happy. When you were sad, you did better, but when you were sad, you did worse on diminished verbal word associations. So this is an example of the impact of the independent variable on the dependent variable depends upon the other independent variable and which level you're experiencing there. So now we're going to talk about some examples of multifactorial designs. Now we're going to spend some time really delving into great detail into a couple of examples of a within subjects multifactorial design and a between subjects multifactorial design. So we're going to utilize the Adams and Kleck study we've been talking about as an example of a 2x2 within subjects design. So remember that we call it a 2x2 because there are two independent variables, and in this case they each have two levels. If there were three levels of one of them, we'd call it a 2x3. This example I know can throw you a little bit because it's a 2x2, but we look at that, we can interpret it to understand a little bit about the design just based upon that simple formula. they theorized that the eye gaze direction and the facial expressions represent two distinct and isolatable features of the human face. So they were saying that maybe this is really important to understand both of these factors in conveying social information. So according to their theory, what they were saying is eye gaze direction and facial expression can be understood in terms of what they call approach avoidance motivation. This idea that when someone is expressing an emotion to me, sometimes I need to pay attention to what's then going on in the environment, especially negative emotions. And I need to understand, am I the transgressor? Am I the target of this emotion? Or am I simply observing this person, seeing it elsewhere? As you can imagine from an evolutionary perspective, if someone looks at me and they're angry, that means something in a social context. Very different than someone looking away from me who's angry. Maybe I should pay attention to where they're looking, right? If they're looking at me, perhaps I need to consider, is this a fight or flight situation, right? From an evolutionary perspective, we're talking now. If they're looking at me with fear, that means something very differently than someone looking perhaps over my shoulder or to the left at something in a fearful way, because that might represent a threat that I need to make myself aware of. So this is part of that approach avoidance motivation theory that underpins it. It has an evolutionary psychology background. So they have to operationalize those two independent variables, human facial emotion expression and then the direction of eye gaze. So they design a 2x2 within subject's experimental design. Every participant experiences all possible conditions of the independent variables. This facial muscle contraction is the idea that comes from the Ekman research that says we can simulate a easily identifiable, socially communicable example of a human emotion by those specific operational ways of arranging our face. And so what they did is they used anger and fear as their two examples. And then, of course, the gaze direction was direct, looking directly at the camera, therefore at the viewer of the picture, or averting your eyes away. And it's formulated as a within subjects because, like I said, every participant experiences all four conditions. They created the experimental stimuli very much like the example I showed you early in this lecture that basically has these four types of faces: anger with a direct gaze, anger with averted gaze, fear with direct gaze, and fear with averted gaze. As I mentioned, these are actually these same pictures. So they've kept everything as consistent and reliable as possible. All they did was digitally alter the point of the eye gaze in the pictures. So this is the experimental task that they came up with. The person comes in, they go through the consent process, they agree to participate, they sit down at a computer monitor, and they have been instructed on how to label the faces. Usually it's like 2 keys or like 2 buttons, and you're told, all we want you to do is to tell us, is this facial expression fear or anger? Just as quickly as you can recognize it, push the buttons. And what they found, and then they're presented with those faces. So they had 32 participants, and each experimental task included 240 slides. Okay, so there were 60 examples of each of those possible 4 conditions. Okay, 60 times 4, 240. And they had them essentially answer that question, fear or anger, as fast as they possibly could. What they found was that participants were faster at making correct judgments about anger from the anger faces with direct gaze, and were faster to make correct fear judgment for fear faces with an indirect gaze. Now let's think about what that means in terms of the theory, the approach avoidance. As I mentioned, from an evolutionary perspective, as you can imagine, those 2 particular circumstances are quite important in terms of survival. Someone looking at you with anger in a direct way is a potential threat. Someone looking away from you with a look of fear is potentially also identifying a threat. So the idea is that from an evolutionary perspective, the way that our brains have evolved, we are a little bit faster, at least statistically significantly faster at those 2 conditions than we are at the opposing 2 conditions, which is fear looking directly at us, which it doesn't pose necessarily a threat. One would assume if someone is immediately fearful of us, we could debate the nuance of that, but you can understand what I mean. Or someone looking away from us in anger, because it doesn't directly relate to us at that moment. So how do we interpret outcomes in a 2x2 design like Adams and Kleck? So this is a multifactorial 2x2 design, and then they have those, as I said, the 2 independent variables, which we've already covered. There are two basic ways in a multifactorial design that we can examine the impact, the hypothesized causal effect of the independent variable manipulation on the outcome on the dependent variable, and those are called main effects and interactions, and we've already touched on this a little bit. The main effect is essentially the isolated influence of just one independent variable on the dependent variable. You essentially collapse the other condition into one large group. Okay? And you only distinguish it based upon which condition it is for independent variable 1. For example, you collapse independent variable 2 in this case. Then you can do the same, right? You can collapse across all conditions of independent variable 1 and look at just the main effect of independent variable 2 on the dependent variable. And we often characterize this 2x2 design as a matrix, so you've got sort of rows and columns on an x and y axis, which we'll look at in some detail in a minute. So the means across the row is the impact of one independent variable, and the averages of the means across the other is the impact of the second independent variable. Excuse me. The more interesting thing, I think, of course, one of the things that's so beneficial about a multifactorial design is we can also look at interactions. And so this is, an interaction is a statistical interaction when the effect of one level of the independent variable depends on the particular level of the other independent variable. And so, as I said, Adams and Kleck found a strong interaction for this idea. There is an interaction between the two independent variables' emotional expression and gaze direction, but that it's really, we think, based upon evolutionary theory, why we tend to be faster at identifying those two conditions that pose potential threats to us in that sense. So let's look at an example of this, and this is all from your book, from their discussion of this. Again, we have the anger, we have the fear. This is our independent variable B. Independent variable A is the direction of eye gaze, right? And we have over here the means for each row. These are response times, I believe, in milliseconds. And so as you see here, if I look at the means across here, let me get my little pointer so you can see that thing. All right, so if I'm looking at this 888.2 and this 917.9, what I'm looking at is the main effect for anger versus fear. And what we find is that people are faster. Now, I don't know if this is statistically significant or not based upon this little graph, but essentially we see that you're a little bit faster at recognizing anger than fear. That's called a main effect for independent variable B. If we look at these down here, we find that there's not really any main effect for eye gaze. These means here. They're very, very close, right? So the first interpretation of this is there seems to be, we have to look at the statistical test, or the inferential stats that tell us whether or not it's significant, but there is, at least by number, a noticeable difference between anger and fear, but no difference between eye gaze alone, because I'm collapsing across anger and fear for eye gaze. So, as I said, we have two distinct sources of information. We have the main effect and we have the interactions. And the main effects are statistically independent of interaction effects. You can have no main effect, which we're going to look at an example of that in a minute, and still have a significant interaction, or vice versa. You can have two statistically significant main effects for each variable, but no interaction whatsoever. They can just sort of operate independently of one another to have an impact on the dependent variable. So to interpret an interaction, we have to look first at the simple main effects. We have to look and see. And so in that last case, in the last example, there was apparently, there appeared to be at least, a main effect for anger versus fear, for the type of emotion, but not for whether or not the person is looking at you or away from you. Now, as we know, there's an interaction that explains the relationship between those two independent variables. Typically, one of the ways we do this is by just graphing this data. By visually looking at it, it allows you to visually interpret a couple of important factors. And we use line graphs of the simple main effects. And we're going to look at a couple of examples of these in a moment. As I said, the simple main effect is really just the whether or not there's a independent response for one independent variable by itself. There are various types of visual cues we look for when we're interpreting a graph, this type of line graph. Sometimes an interaction is referred to as a crossover interaction, which, as you'll see in a moment, is because the lines that we graph are not parallel. They cross over one another, sometimes kind of like in a very classic X pattern. Sometimes they sort of graph out and they will eventually cross one another. Either way, it is what's called a crossover, or just simply an interaction between those two main effects. There's also what's called an antagonistic interaction, which is where the independent variables can show opposite effects from one another. And then, strangely enough, they can kind of cancel out if you're, and so there's no interaction in a certain sense, but they can be affecting the dependent variable in opposite ways. As we said, in this case, there was an antagonistic interaction that direct eye gaze led to faster responses to anger faces, but slower responses to fear faces. Averted gaze led to faster responses to fear faces, but slower responses to angry faces. So exact opposite interaction in that sense. If our two main effects are parallel, roughly parallel, then that indicates that there's no interaction. We call this additivity. They may contribute to one another, but they're not interacting in any significant way across the levels of the independent variable. So if you see parallel lines graphed, then we essentially think, well, there's no interaction here. If you see two lines that are lying right on top of one another, then that means that there is, essentially no interaction and there's a similar effect. If there is two horizontal lines that overlap each other, almost to the extent that it appears to be one line, that means there is no measurable or distinguished relationship between the independent and the dependent variable, and this is the null hypothesis. We also call it the null effect. If you see two parallel lines that are lying on top of one another, what you essentially have is that all four conditions pretty much look exactly the same to one another. Let's look at some examples. Okay, so now this is a graph of just the main effect, right? This is just the main effect for eye gaze direction. As I said, those numbers are quite close to one another, and as you can see, this is nearly a horizontal line, excuse me. So this would be no main effect. This is the main effect for response time related to which emotional face you were seeing, anger versus fear. As you can see, this line has a noticeable slope that is not horizontal. The response time for anger faster than the response time for fear, about 917 to 888, I believe it was. So this is a main effect. Because the line is not flat, we would interpret this as a main effect for the type of emotion independent variable. Now, this is what happens when we graph both. If we graph both of the main effects, right? What we see is this crossover interaction. So for averted gaze with anger versus direct, you see that main effect. For direct versus averted, for fear, you see the opposite effect. And so this is why we visualize this as an interaction, as a crossover. So this is some fake data I want to present. This is basically, this is just examples of the same cells we were looking at a moment ago with the real data. And now we're just going to look at some examples of how we would interpret this sort of, these fake situations, right? So in this case, as you notice, 800 milliseconds in all four cells. Therefore, the means are the means are 800, the means are 800 here. That would look like this. Now, what you can't see here is that there is another line laying on top of this one. We have a horizontal line with, and both of the main effects are lying on top of one another, so we can't even distinguish it as more than one. This is a null hypothesis, no effect of either, no interaction either. Now let's look at this example. In this case, the mean in anger is 500 for direct gaze, 530 for averted gaze. In the direct gaze for fear, it's 600, and the direct gaze averted gaze for fear is 630. What we get then is some different means. And in this case, you look at this and you say, well, fear and anger, there seems to be a main effect for independent variable B. There's a significant difference here between these two numbers, right? I'm using that term loosely. I don't know the actual p-value for this, but this is visually, it appears to be different. We'd have to run a statistical test to say for sure if it's statistically significant. And then as we look across the columns for the main effect for direction, it seems that averted is faster to be interpreted compared to direct. Now let's look at what that graph would look like. We now have two parallel lines, right? This is the line that represents anger, and this is the line that represents fear. This is a design that yields a main effect for emotional expression Fear is definitely slower to be responded to than anger, but no effect for eye gaze because it's a flat line, direct to averted, it's a flat line, and these lines essentially are not ever going to cross over one another or very nearly to it parallel. Therefore, we would say there is a main effect for emotional expression, but no effect of eye gaze and no interaction. This is an example of some data that would say that there's a main effect for eye gaze and emotional expression. Both of these lines have a slope to them that go up, right? But no interaction of the two. Again, they're parallel. Let's look at some other fake data here for a moment. This is one that gives us an interaction of the two, but no main effect for either. So if you look 500, 600, 600, 500, we're seeing a reversal here, right? But those means come out as exactly the same in all four of these situations. And therefore, we would say no main effect for emotional expression, independent variable B, no main effect for direction of eye gaze. But here's what's interesting when we look at that graph. So we have zero main effects, but we do have a significant interaction between the two variables. That's how we would interpret these lines crossing over one another. So we call this, 2x2 logic. Let's think about this and what this means in terms of our interpretations. They found some really important things. They found a significant effect for facial expression. Anger and fear are interpreted at different speeds. They found a significant effect for the interaction of facial expression and gaze direction. As you remember, those are those two cells that stood out. But by contrast, they didn't really seem to see any main effect for gaze direction itself. So we might use terms like additivity to indicate the absence of an interaction between independent variables. Additivity and no interaction are synonymous terms. Okay, so now that we've explored an example of a within subjects multifactorial design, let's talk about a between subjects example. So if you remember, in a between subjects experiment, each participant only experiences one level of the independent variable. In a multifactorial design, what that means then is that each participant experiences only one condition of those combined variables. So when you create that cell in a 2x2, for example, you have 4 conditions. That means you're going to be in one of those 4 conditions as a participant in that study. So just to reiterate the nomenclature for a moment, if you had a 2x3 between subjects design, you would have 6 combinations of the 2 independent variables, or we might call these 6 experimental conditions. Conditions is a term we're going to use to kind of represent each of those unique cells, that unique combination of these multiple independent variables. So participants would then be randomly and exclusively assigned to one and only one experimental condition, and then you would compare their responses on a dependent variable across those 6 separate groups of subjects, I should say. In the 2x3 between subjects design, for example, we can talk about research in your book by Baumeister, Twenge, and Nuss from 2002. And this is where they staged an event that manipulated the effects of social exclusion on participants' GRE scores. We're not going to get into all kinds of details about this, but it was an interesting study. They were essentially told things about their future. It was staged. They were informed, hey, this is just part of the experiment. You're going to be informed about something about their future. And they were told either they would be alone, which is called the future alone condition. They would have a rich and rewarding relationship, the future belonging condition, or that they would be accident prone, which they call the future misfortune condition. And so participants were told that their future forecast information was bogus at one point or another, right? But that's the other manipulation, is what they call the other condition is where they told before or after they answered a series of GRE questions. So and then what they would do is within subjects design, for example, would not work in this case, right? Because you can't have someone told both before and after that this is bogus. You can also not have someone told that they're going to have all of these conditions or multiple conditions and have it not muddy the waters and pollute. So for example, it's difficult to tell them you're going to be alone and you're going to have a rich and rewarding relationship. That wouldn't make sense logically. So they basically created this condition as to whether or not you heard about it. You heard, oh, by the way, this is totally bogus. It was part of the experiment. There was a debriefing, essentially. Did that happen after answering the GRE questions? They call that the recall condition, or before answering the GRE questions, which they call the encoding condition. So therefore, you have two variables, right? You have which is the bogus future that you're told about, and when is it revealed that it's bogus, hence 3 by 2 or 2 by 3 in this case. Um, because this was a between subjects design, it required more participants. It had an N of 65 people. Um, and the researchers used random assignment in order to minimize the possibility of group differences before the manipulation. What they didn't do is what's called a manipulation check, but this is something that you can do, which is that you make your random assignment, and you then go and you have some questions that are directed at the participants to determine whether they objectively believed the fake feedback to begin with. So you kind of figure out, did my manipulation, you're checking on my manipulation of the independent variable. So how do we interpret a between subjects experiment in a multifactorial design? Well, we're still using the idea of main effects and interactions. Um, so they tested for the main effect of and interaction effects of social connectivity, and then when you were told about this information. And they predicted a statistical interaction in which the effects of social exclusion would affect only those participants who took the reading test believing they would end up alone in life, meaning that being told that fortune, and then of course potentially being affected by it before being told it's bogus, you still had to carry out the task. Well, then they thought that would have a negative impact on people's performance on these GRE questions. What they found was the effect depended exclusively on the interaction of aloneness and having to recall information to answer questions. So again, you examine statistical interactions by graphing the simple main effects and seeing how those lines look in relationship to one another. Any main effect in a multifactorial experiment of two or more independent variables should only be considered after you've examined for interactions. That's an important concept, and I think it's something that's hard to wrap your head around logically, but if the impact of a single independent variable is dependent or is related, if it interacts with a particular condition of the other independent variable, then as you can imagine, you don't really interpret main effects. You can run into a problem there. For example, going back to the previous study we were discussing, eye gaze and the emotion being expressed. In an interaction, you might look at that data without studying the graph, and it would look like it was a wash. But in reality, what you have is this more complex interplay. There's no simple main effect, for example. There can be no simple main effects for either of them, but still an interaction. So ultimately, the first thing you do in interpreting the data in a multifactorial design between or within subjects is you look for the interactions first. If there is no interaction, then you simply just go to interpreting main effects. So what they found in their study, in this example from Baumeister and colleagues, they revealed a statistical interaction between social connectivity and intellectual thought. So it was related to recall of being, alone later in life and then having to go through that process of answering those GRE questions. That particular condition was the one that showed a relationship or interaction between the two independent variables. So this is the data from that study. Of course, remember what we said. This is columns and rows, right? We have independent variable A, which is encoding or recall, right? Did it happen before or after you had to take the GRE questions? And then you have the three levels of independent variable B: future alone, future misfortune, future belonging. So these are all the individual cells. These are the 6 cells here, right, that are created by that 2 by 3 combination. These are the means for encoding versus recall. What they found was that people that were going into the situation with any sort of fortune being predicted tended to do worse. On average, it's going to be a main effect for encoding versus recall. And then if you notice, future misfortune and future belonging are very similar to one another in this main effect, but future alone is significantly worse. So this is where we would examine main effects, but until we graph it, we don't really see the interaction. And this is where you notice that these lines are, they have a separation of slope, but they're very close. And then you have this dramatic effect for future alone. So this is where you recognize the interaction graphically. Okay, last thing I want to talk about in this chapter is what sort of statistical tests do we use to test our hypotheses when we're using multifactorial designs, right? So these are all various types of inferential statistics that we generally use to get, to find a statistically significant relationship between our independent and dependent variables. The one most commonly used in a multifactorial design is called an ANOVA, or an analysis of variance. So ANOVA is just an abbreviation for the term analysis of variance. It's also sometimes referred to as a one-way analysis of variance, and this is when you're comparing three or more groups of numbers, right? So when you have more than just a simple two things you're comparing, one independent variable and one dependent variable, as soon as you introduce a third factor, in this case another additional independent variable, we utilize an analysis of variance. So if we're comparing only two groups, like for example, A and B, right? If we only had one independent variable with two conditions, then we can utilize something called a t-test, which is an inferential statistic that compares two groups only, two and only two groups. As soon as we incorporate more than two, levels of that, or more than one independent variable, excuse me, and we run into essentially three groups now at least to compare, we have to, we said, well, maybe what I'll do is I'll just run three t-tests, right? I can compare A to B, A to C, and B to C, and I'll cover all my bases. The problem is that when you run multiple significance tests, what happens is you are accumulating the chance of making a type 1 error, of essentially having a false positive. Remember, a type 1 error is when you falsely conclude there's significance when there's no real effect present. It's when you incorrectly reject the null hypothesis, the hypothesis that there's no relationship between the variables. This is called alpha inflation, and it's a concern when you're running t-tests. So the idea is that you need to run a single test that tells you whether any of the groups are significantly different from one another from a statistical perspective. So to try and avoid alpha inflation, we can run what's called an ANOVA. So that's what this test is. So the ANOVA is exactly the kind of test we need when we have more than two groups that we're comparing to one another. It does not look at the differences between pairs of group means. It doesn't compare A to B or B to A or C to A as you would with three independent simple t-tests, for example. It looks at the entire collection of all the means across all the groups, all the conditions, the experimental conditions, and compares how much they spread out from one another compared to how much spread there is within the groups. So this is an important concept I want to stick with for just a moment. We know that our individuals who are participants bring variability, right? They bring certain individual characteristics. They're different from one another. We know that. Human beings vary across a lot of these things that we're studying in a meaningful way, usually in a normal distribution. Most people are close to average and the outliers are rare. By virtue of that, we know there's going to be some variability within every group, every condition that we assign people to. What we want to look at in an F-test is the amount of variability between those groups greater than what you're seeing within the groups? If it is, then we can assume that there is some significance in what we did in the manipulation of our independent variable that had an impact and affected the dependent variable above and beyond whatever normal variability people brought to the table. So if you run your ANOVA, it simply tells you if there is significant differences between at least one combination of groups within there. It doesn't even tell you which one specifically. But if it reaches that alpha level of 0.05, for example, that you may set, then you can conclude that these groups are not the same in a way that's statistically significant. The means varied from each other by too much an amount to be accounted for just by individual variability of people within those groups. And you can then go back and run what are called post hoc tests to figure out which specific groups differed from one another and how. But by running just the one ANOVA at first, what you're doing is you're avoiding that inflation of type 1 error, or that alpha inflation that comes with just running every possible combination. As you can imagine, a 2x3 is six different groups. You have to look at different means, and it starts to create that factorial effect we discussed in the last chapter, where you've got lots of different comparisons to run, and you're just making more and more possibility for yourself to have a false positive. So the ANOVA is a way to run an initial test that may say, nah, there's no difference at all, and therefore I don't need to go any further. But if there is, then I can delve into it a little bit deeper and tread a little more lightly and try to avoid that alpha inflation. So as I said, I don't want you to understand all of the statistics in great detail for the purposes of this class. I just want you to know what an ANOVA is and what it does in terms of looking at a multifactorial design, that it's the appropriate inferential statistic we would use to test significance in a design that has more than two groups that we're comparing. So to sum up a little bit and conclude, multifactorial designs are just adding another layer of complexity to what we talked about in the previous chapter. You have an experimental design that's a single factor design that has one independent variable. What multifactorial designs do is they allow us to bring in another construct, right? To bring in another player in whatever relationship it is that we're studying. And you have not only the ability to see if there's a direct main effect between each of those independent variables and this outcome, this dependent variable you're interested in, but now you have this additional tool, this really fascinating thing of looking at how they interact with one another potentially. As we've alluded to, human behavior is complex. Being able to introduce complexity and nuance is good. It means that we can test theories that are themselves more complex, that incorporate more than single factors. And so hopefully what we're doing is we're approaching increased validity of our study in terms of actually measuring what we say it is we're measuring and getting a good handle on how these hypothetical constructs really do work with or against one another in the real world.